Welcome to another screencast where this time I'll be discussing some characteristics of proteins. And so by the end of this, hopefully you will be able to identify and describe here the monomers of proteins, that is the individual single units, mono. Some examples of proteins, the structure of an amino acid. An amino acid, again, is the monomer, so it's important to know the structure of the amino acid there. I didn't write it on here, but we also just want to make sure that we know the elements that are involved here with proteins. Then identifying and describing here the levels of structure of proteins, and then any sort of enzymes, and there are a lot of them that are associated with protein metabolism. And these enzymes are going to be named for sort of the examples here of the structures of the amino acid and the structure of the polypeptide of the protein. Right? So this is what I mean by the levels of structure. Hopefully you'll see that in just a minute. Describe here and explain the metabolism of the proteins and then the importance. Again, this is always what we want to get to, the importance of proteins or amino acids to cells. What's the big deal? Who cares about that? So here we go, our coloring book page and our complex food substances. We're going to be talking over here about the proteins. And proteins are also sometimes referred to as polypeptides. Right? So you'll hear me sort of flip back and forth between these terms here sometimes, polypeptides and proteins. And so we will typically ingest proteins at the polypeptide form or the oligopeptide form. And you see that these are, just by this coloring book structure here, these dots, these little circles here that are all bonded together, implies that that these are polymers, right, made up of individual little subunits right here. So these oligopeptides, and again, oligo would be few of these little circles together, and then polypeptides would be many of them on the order of thousands of these little individual units here. We can break those down into, chemically digest those into dipeptides, little dimers right here, but then ultimately we want to get down to digesting them down into the individual amino acids. And again, each individual circle here represents an amino acid, because that amino acid is what is then absorbed into the bloodstream here and then transported this way to the cells. Right now, in some cases we actually will ingest amino acids, but the majority of protein that we ingest here is in the form of this oligopeptide or polypeptide level structure. So let's take a look here at what an amino acid is. So an amino acid is the monomer of a protein, and from this we can see that proteins will have carbon, hydrogen, oxygen, and nitrogen, and then to some degree have sulfur as well. So carbon, hydrogen, oxygen, nitrogen here. Here's the amino acid. This amino part is because you have this NH2 grouping right here. The acid part refers to this carboxylic acid over here, this C with a double bond O and this OH right here. AP biology and AP chemistry kids are a little bit more comfortable with this right now, but suffice it to say that this amino acid has this nitrogen-carbon backbone to it, has this hydrogen sticking off the central carbon here, and then this is a variable group, or known as R. Is an R group and is variable, and there are 20 different R groups there, and this is the table of all those different R groups. Right, some that make the amino acid polar, some that make them non-polar, some that make them electrically charged. By no means do you have to memorize this. I'm just showing it to you as an example, right, of what the 20 different amino acids are right down there. So to get a protein, though, you're going to take these amino acids and string them together. You're going to make these peptide bonds between the carboxy end here and the amino end of the other amino acid. And so again, you end up just stringing these amino acids together to create many peptide bonds, which is where you get the term polypeptide from. Right? And so here, this would be a dipeptide. Right? And you have two amino acids, right? And then this would be a tripeptide. You have three amino acids together. And if you keep stringing amino acids together, you eventually get a polypeptide. Right? And so you get this, that's what this is supposed to be showing you right here, where each one of these circles is an amino acid, right? And they all have their own specific name, so each circle is an amino acid. And the string of amino acids represents the primary structure of the protein. And then what happens is this begins to fold. in either beta pleated sheets or alpha helices. And again, AP Bio people are a little bit more comfortable with this. It doesn't really matter, just as long as you understand that the proteins are rather large structures and they result from multiple levels of folding. Here's the tertiary structure right here, all the way up to then this quaternary level structure. Right? So for example, a protein collagen that we've seen in connective tissue, right? This is the fibers in connective tissue. And so this collagen is actually 3 polypeptide chains all woven together. So if I was to zoom in right here, right, what I would see ultimately would be this string of amino acids all chemically bonded together. Right? And much the same way that polysaccharides have different characteristics than the individual monosaccharides, these polymers, these polypeptides, have different characteristics than each individual amino acid. Right? Down here we see a couple of other examples of proteins. Here's actin and myosin, and these are the 2 main proteins found in skeletal muscle, right, as well as cardiac and smooth muscle. But when we eat muscle, if we consume it, then these are some of the proteins that we're consuming, as well as some of that connective tissue, etc. Right? So these proteins typically are large structures, right, that need to be, of course, metabolized and broken down. So again, the proteins, these oligopeptides, these polypeptides, would be things like collagen and actin and myosin that we are consuming. And there are quite a bit of different proteins that we consume. But again, the important part is that they're all going to need to be broken down into these individual, amino acids. Whoops, each individual amino acid right there, because that's what gets absorbed into the bloodstream. Those are the monomers. So what are the enzymes that do that? Well, there are a lot of them. Right? Pepsin is one enzyme that is in the stomach, and that is going to be breaking down the individual proteins into larger polypeptides, right? So you have this chain of amino acids here. Let's say that each one of these represents a couple hundred right now. That pepsin breaks that into maybe 2 smaller chains here. These are larger polypeptides, right? And then we have these whole slew of enzymes in the pancreas that are released by the pancreas. Trypsin is one, chymotrypsin is one, carboxypeptidase is one. These are all going to be breaking down the proteins, those polypeptides, into progressively smaller and smaller chunks. Right? And so ultimately then you get to the enzymes that are on the brush border, and these are amino peptidases, carboxypeptidases, dipeptidases, right, that are maybe complex in name, but if you look at what's similar about them, right, peptidase, they're breaking these peptide bonds between the individual amino acids. So again, that would be a peptide bond. There's an amino acid, peptide bond, amino acid. These enzymes are breaking these peptide bonds, and so you end up getting individual amino acids, right, that then enter the capillary blood at the villi and will be transported in the bloodstream. So again, the majority of those enzymes are working here in the small intestine and are derived from enzymes that are either in the small intestine or that are released from the pancreas. Right? So again, there's another visual. And finally here, who cares about proteins or amino acids? Well, the cellular uses here, let's start at the bottom and work our way up. Right? Structural proteins, connective tissue, so your cells will use the amino acids that you take in to build their own, to rebuild their own polypeptides that are needed, right, for, again, connective tissue, muscle are some examples. You also need amino acids to make the enzymes, right, and antibodies, right, hemoglobin we talked about as well, and so these are functional proteins as well as sort of structural proteins. So why do cells need them? Structural and functional uses. But then lastly, we can in desperate times use amino acids. If the essential amino acids are lacking, we can use ATP there for energy sources, right? So we can use these amino acids in cell respiration and in the different parts of cell respiration there to produce ATP. So that is sort of the third use, but this is kind of the last ditch effort there, right? Is an energy sources for that. Okay, so hopefully that is helpful here. You're able to talk a little bit about those monomers of proteins, and again, those are the amino acids. And knowing the structure of the amino acid and the elements involved there is important. Give me some examples of proteins. This level of structure, I'm not really super worried if you didn't get that, but you can go back and take a look at that again. What is important though are the enzymes that are associated with protein metabolism, and we talk a little bit more about that when we do the actual physiology there of the small intestine and the pancreas. And being able to, again, just generally talk here about the metabolism of proteins and the breakdown, like how it goes from polypeptides to oligopeptides to dipeptides down to amino acids. And then finally here, the importance of the proteins or amino acids to cells, right? And again, they have three roles there in cells in terms of structural, functional, and then energetic properties. Okay, thanks for listening. Hope that helps you bring your questions to class.